Hello, so my assigned protein along with this mutation was with Piezo 1, and it's where the histidine replaces the arginine, and I believe the scientific, I don't know how to say that, like the name was, the mutation was R2456H. And so basically Piezo 1 is a mechanosensitive ion channel, and it is a physical receptor that responds to pressure and stress of fluid in the body. It is essential for bodily processes such as blood flow and the detection of changes in blood pressure. I'm choosing to categorize this as a receptor. A receptor is a protein that detects specific signals to trigger a cellular response. They can either be chemical, or in the case of Piezo 1, they can be mechanical. That is why it's referred to as a mechanoreceptor. Piezo 1 is normally found in the plasma membrane. It is present all over the body in many different types of cells. And those cells include epithelial, endothelial, neural, osteoblast, bladder, and red blood cells. It is a human receptor, so it is present in humans and organs like the skin, lungs, bladder, colon, and kidney. So pretty much Piezo 1 can be found anywhere in your body. In the case of this mutation, the protein actually becomes more active. Normally the ion channel would open when the cell membrane is stretched or under pressure, allowing ions like sodium and calcium to flow through, and then close after a short period of time. With the mutation, however, the channel either doesn't close at all, or it stays open for longer than what it needs to, which causes excess ion flow, which leads to cell dehydration. All right, so this is my model, and here, let me show half of it first. So this is like the plasma membrane, and like I said, the Piezo 1 receptor is actually embedded into the plasma membrane. So these are like the hydrophilic heads, and like this represents like the hydrophobic tails. This is a depiction of a plasma membrane, and then these are like the three blades at the top. And then yeah, so the receptor just opens and closes as a response to changes in pressure, and it allows the flow of ions through, and then after a while it just closes back. So with the regular Piezo 1, like no mutation, the guanidinium group gives the side chain hydrophilic properties. Another functional group that's present is amino groups, and those are also hydrophilic. The side chain is positively charged. Arginine has one of the largest side chains, so I would consider it to be on the larger side out of the other 19 amino acids. Since arginine is positively charged, it can form ionic bonds with negatively charged side chains. These bonds can help stabilize the protein structure. The guanidinium group has multiple nitrogens that can either donate or accept hydrogens, so this allows many bonds of other molecules to bond to arginine through hydrogen bonds. Acid side chain is hydrophilic. It is usually either neutral or positively charged. I would consider histidine to be on the medium or a smaller side. Unlike the original arginine, which is very positive, histidine is positive and can be neutral, as said before. Due to this, it has the ability to form ionic bonds. However, they are weaker and not as many interactions are formed. Histidine also has the ability to form hydrogen bonds similar to the arginine through its imidazole functional group. I don't know if I said that right. Just like arginine forms hydrogen bonds through its many nitrogen atoms. However, with the histidine, there are less binding sites than arginine, so the overall interactions are weaker. This is what causes the channel to stay open longer, allowing an excess of calcium and sodium ions to flow through. Hence, what causes the water to leave the cell, osmosis, leading to dehydration. So in humans, this causes a person to develop hereditary xerocytosis, which is a type of hemolytic anemia. Some side effects of this are fatigue, pale skin, jaundice, and an enlarged spleen. And yeah, so that's all about Piezo 1. Some limitations about my project are obviously with the model, the size doesn't match up with how it is in our bodies. Another limitation would be with the materials used. It's hard to properly depict how it actually works, like how it actually moves within our body. It's also hard to depict how the ions flow through. Like, it's hard to show that because it's a 3D model, but it's like not really like 3D. Like, it's hard to show everything. Yeah, so the materials used are also another limitation. Obviously, our plasma membrane is not shaped like stars and pipe cleaner. But yeah, so learning about this mutation though really helped me connect the dots on how molecular biology and human health really go hand in hand. It shows how even tiny changes at the molecular level can impact the whole body, emphasizing the importance of studying biomedical research, future biomedical engineer, and improving human health.